Welcome to lesson 16. I'm re-recording this lesson because the initial recording that I had when I went through this with you on Zoom didn't work because I missed out on the fact that I couldn't hear you because I had the volume turned down, which was a bit of a mistake. And so instead, halfway through the lesson, you can hear Brendan having a conversation with Alethea about her birthday. Happy birthday, by the way. So what I'm going to do today is I'm going to quickly talk to you guys about DNA structure, and you guys can have a recording of this. You can pause it wherever you want and go back and talk about it. So let's talk about this and we'll make our way through. We will be able to explain how DNA is shaped and also how base pairs match. Now, we haven't talked about what a base pair is. We've talked about DNA though. This is a bit of a different look to what we've been doing. Let's go back a sec to see how what we've been looking at so far. So far we've been looking at DNA as, what it does. It's a blueprint that gives instructions to proteins to then make a copy of a organism. We said it wraps itself up. So DNA wraps itself up into these tight little bundles. These tight little bundles, they're called chromosomes. We've got 46 of them in our body, and the chromosomes are what goes inside the cell, inside the nucleus, and the nucleus is inside the cell body. Now, this is kind of interesting because what we're going to be doing is we're going to be going the opposite way. Instead of getting into bigger, we're going to go smaller and look at specifically what makes up this DNA structure. We also talked about the different types of chromosomes. We said on Wednesday we've got two types. We've got autosomes, which are the regular chromosomes, and then down the bottom, just above my head, we've got these sex chromosomes. Now, the sex chromosomes are broken up into two types: X chromosomes, which means you've got a girl, and Y chromosomes, which means you've got a boy. You've got an X and a Y, that means a man can give an X and a Y or a Y to their children, which will change the sex of the child. So what do we talk about when we talk about how DNA is shaped? So DNA is a molecule. Now this is, and it has multiple parts. A lot of students forget that DNA is not an atom, it's not a cell, it's not a nucleus, DNA itself is not actually alive. It is just a complex molecule. Now, it's made up of different parts, but these parts ultimately have a structure called a double helix. Now, the reason we call this a double helix is because ultimately it has two rails, just like how a ladder might have two rails. And it has rungs. These rungs go between the two different rails. The reason it's called a helix is because it's actually a curved surface. Now, a single helix would look like this. And in fact, helix actually comes from the word, helical, which is like a spiral. So a single helix would have just one. A double helix is like, imagine you've got a ladder, but you instead twist this side so it comes over here. And then you twist the other side so it comes over there. And then, let me just try that again. You twist the other side so it comes that way. And so therefore you end up with a DNA strand. A triple helix would actually have three shapes that would then twist. But yeah, we're not interested in triple helixes. We're just interested in the double helix, which is DNA. Okay. What I do want you to copy down on this slide is just this section here, by the way. It's just the DNA molecule and how it's made. It's made by rungs on a ladder, and it's a twisting ladder. It actually takes a normal ladder and we twist it. So, you can sort of maybe, even if I just do this. So we take a normal piece of paper and we twist it around. That's what we're talking about when we talk about a helix. Now, what we can talk about now is, what about the zoomed in structure? These rails that are going around the outside actually have a structure of their own. What we refer to these rails is, we refer to these rails as a backbone. Kind of like, how humans and animals have backbones. We refer to the DNA as having a backbone. And the backbone is made up of two smaller molecules. Remember I said DNA is a complicated molecule. It's made up of two smaller molecules. One of them, and these molecules repeat, and they are a phosphate unit and a sugar. Here's the sugar, and here's the phosphate. The sugar, by the way, is actually called oxyribose. So that's where the deoxyribose comes into play. Deoxy, by the way, meaning two oxy. So deoxyribose is the name of the sugar. Now, these parts repeat over and over and over again. So it's going to go phosphate, sugar, all the way down the molecule. And they're always going to be the same phosphate and the same sugar. Now, there is an extra part here. The extra part here is this, called a base. Now, a base is just like how we would normally refer to acids and bases. That's what it is. Now, they can be a little bit special, and I'm going to talk about why these bases are special. But at the moment, we're just going to accept it's called a base. Now, what we can then do is we can then say the three molecules together, the phosphate, the sugar, and the base, make up something that's called a nucleotide. So we have, this is a nucleotide. And I want you to copy down this side of the page here, over here on the left. So over here on the right, we have a more complex thing. And we can see the pattern here: sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And actually, the same pattern goes on the other side as well. It's the same thing that keeps repeating itself. In the middle, the rungs of the ladder are made up of these bases: base, base, base. Now, here's a question that I've got for you. And I think I asked this last time, but I'm going to ask it again. How many nucleotides are there? Well, each nucleotide has one sugar. So, if each nucleotide has one sugar, we can calculate this by going 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Each nucleotide has one base. We can see that there are 12 bases. There are 12 nucleotides. Now, what is interesting is there's only 6 pairs though. 1, 2, 3, 4, 5, 6. So there are 6 pairs. So that's going to be important in a second. But let's just talk about that. Let's recap this page. Ultimately, it's the backbone of DNA is a phosphate sugar backbone. They keep repeating over and over and over and over and over again. The base is attached to the sugar. And this occurs on both sides of DNA. Altogether, the phosphate, the sugar, and the base is referred to as a nucleotide. Now, these bases are a bit special. Let's talk about these bases now. This is thymine. There are 4 bases that make up DNA. And these are the 4 bases that are in all living things. Those bases are adenine, thymine, cytosine, and guanine. Now, I'm not going to expect you guys to really memorize what these names are all the time. All I really want you to memorize is A, T, G, and C, or A, T, C, and G. I've got some basic image of this. Obviously, this is not what they look like. They're actually a complicated molecule. But yeah, these are the 4 bases, and this is the alphabet that DNA uses. Now you might say, well, you can't write many words with just 4 letters, but with different orders, different sequences, you can actually create enough complexity to make the genetic code. So, how does that work? These bases have pairs, and they will only fit together with their matching pair. A always pairs with T, like here. They join together, and this is now why you can tell why I drew them in this way. And C always pairs with G. The pattern that I've got here is apple tree, so AT, and car garage, CG. This is something I stole from a YouTube video, but I liked it. You can come up with your own memory technique. You might be like cool guy and annoying teacher or whatever. It doesn't matter as long as you've got a way that you can remember them. You can tell that I've chosen these particular shapes for a reason. The T will only ever go with an A. Okay? The T can't even go with another T. So if you took 2 T's together, these two can't go together because they wouldn't fit. You can't force a G to go into a T. They are specific bases that only fit together. So how can we use this to our advantage? Why would we care? We care about this because it helps us make matches. Let's just say we've got, here's our phosphate sugar backbone, and we're going to put half of the DNA strand here. So with just one side of the DNA, you can create the opposite side to match it. How does that work? Well, let's look at this T. All right, what goes with T? I know A goes with T because apple tree. So A goes with T. So we've got an A going on here. C, well, car garage. C goes with G. Well, we've got another T, so there's another A there because T goes with A. A, apple tree. A goes with T. G, car garage. Well, I need a C down here. That has meant that I have now, because I know how these fit in pairs, I know what must go with what. And now I've just completed the other side of the DNA strand. This is what DNA uses in order to replicate. And we're going to talk about this in a second in the next lesson. But this is what they use to replicate. With just one side of DNA, we can create the opposite side. With just 4 letters, we can make a really complex code. You might disagree with me, but get this. With just 1, 2, 3, 4, 5 base pairs, you can write 1,000 different combinations. 1,024 to be precise. You could have A, you could have AT, you could have AC, CTC. You can have all T's or all A's. There are so many different combinations, it's not funny. And that's just with 5 pairs. With 10 pairs, it doesn't just increase double, it increases by a factor of 2 to the power of, 2 to the power of 10. It's going to go, through the roof. But I just wanted you guys to see that I can easily match one to the other. I can also use this information to do some other things. Okay, if I know that, for example, let's just say I've got a DNA strand, and let's just say I tell you, hey, there are 14 thymines. Well, you can straight away say, well, if there are 14 thymines, there must be 14 adenines. Because you know that, well, if there's 14, there's 14. If there is, let's just say there is, let's use a different one up here. In this one over here, there is 10% of them is, guanine. Well, that means that 10% of them will have to be cytosine. And the rest, the other 90%, is going to be broken up into 45% adenine and 45% thymine. And this is important because there was a question that I saw that had this kind of information. But I know I can just break these down because I know if there is a T, it has to bond with an A. And if there's an A, there has to be a T nearby. So let's just go back over really quickly what we've just done. We've said DNA has a structure. Its structure is a double helix, which means it's a spiral shape. It's a spiral that's twisting two ladders. It's twisting two rails of a ladder. Those rails twist around each other to produce a curve. Inside that twist, we've got nucleotides. Each nucleotide has a phosphate, a sugar, and a base. There's three parts. And the backbone just goes sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, over and over and over and over and over again. The base, well, the base is attached to the sugar, and that forms the rungs. It forms the pairs that hold the DNA one side to the other side. Those bases, there are 4 of them. They take the form of adenine, thymine, cytosine, and guanine, or ATCG. We now know that T only fits with A, and C only fits with G. And because we know that these two things only fit together, we can use them to match and create the matching sides of DNA. If you've got any questions, I'm going to leave it there. If you've got any questions, please feel free to email me. I'm more than willing to answer them. Anyway, that's been Macri. Hopefully that helped. I will see you on the next lesson.